Okay. Now, before we actually get into this morning's content then, and we are going to start on, well, we're going to race through the endocrine system and deal with how hormones work. Not in a lot of depth. We're going to sort of do an overview of hormones and then take a few examples. And that will probably only really take us up to break because it's not a system I spend a lot of time on. Partly because one of the options for the assignment is to look at the control of blood glucose and what the hormones are that are involved in that and how they work. So the thing is with this unit, there's a lot of systems to get through, and so a lot of them only get touched quite lightly. So we'll look at the endocrine system in a little while. Just before we start, however, I want to just go over a few points to do with the cells assignment, and I can't see where I've actually kept that from yesterday. No, I don't appear to have saved yesterday's. If I have, I've saved it somewhere stupid, so bear with me. Find yesterday's notebook file. Every now and then there are questions that come up, regardless of drafts and things like that, that I think, oh yes, they're things that I just need to go over with everybody perhaps. And so what I'm going to do is to just pull out a few bits and pieces. Yep, it's not showing me what we did yesterday. We didn't do that much, did we really? We only did that body thing. Maybe I didn't save that at all anyway. I was pretty sure I had, but we'll start a new one anyway. I did list I did actually, I'm sure I saved it because I listed the transport processes, didn't I? So I'm going to take just a very quick ride through what needs to go into the assignment, just to sort of answer any questions and make it clear what needs to be in there. So you start with your introduction, and your introduction starts, if you remember, you've got membrane structure. Then you've got your transport processes, and you start with the bit on the particles, which was things like size, charge, polarity, and concentration difference. And then once you've sorted that out as a basis, you describe the different transport processes that are needed to account for these in relation to the structure of the membrane. So, for example, you've got charged particles that can't move by simple diffusion. If you've got small particles with no charge, they can move by simple diffusion. If you want to get something up a concentration gradient, you need active transport. So you give the different processes and examples of substances that are moved by them. So you go through, I would suggest in the order of simple diffusion, facilitated diffusion, osmosis following that, because it's a particular example of facilitated diffusion really, because you've got the transport proteins, there are specific ones, the aquaporins here, so I'd stick osmosis after facilitated diffusion. And then these are the ones that don't need energy, and then the ones that do need energy, I would, they only really get a couple of sentences each and perhaps diagrams. Active transport and vesicles. Then you have your introduction to the actual practical work. So you might just say, the aim of this investigation is, so you've sort of got the investigation overview comes next. So you might identify your variables and your hypotheses and put your materials and methods in there, all right? So you've got variables and hypothesis. Oh, I should have put aim first, I suppose. So aim there. Then your materials and methods. Okay. And to be honest, if you check that guidance sheet, it's all on there. It tells you what to put where. Then you have your results section. Your results section is the summary of the whole course results. So it's a table course results. So that's 4 rows altogether. All right, your main heading, your water potentials, your mean values, your standard deviations. So table of course results and your graph of course results. And you might at that point want to say, you know, and some anomalies were removed, so it doesn't have to go there. So possibly note anomalies there. They can go in all over the place actually. All right, so you might just say these were calculated with the removal of some. 4 rows. It would be your, at the top it would say water potential. The next row is the values of the water potentials. The next row is the mean values, and the next row is the standard deviations. Yeah? So it's a 4-row table summarising the class results. So top 2 and top 2 and bottom 2 rows off the big spreadsheet. You then have your discussion. All right. Now, you've got various things to cover in here, so you make sure you've got your analysis. These don't have to be identified as separate sections. This should be a continuous discussion. So you identify the isotonic point, which is the water potential of potato. Right, so that's what we're actually trying to find out. You then do extensive comparisons with Kaczynski's stuff. All right, bearing in mind that a lot of Kaczynski's numbers are not his. I've missed an S out of there. Kaczynski. Kaczynski's reporting from way back and a lot of other people's work. So remember, also remember you need to multiply his values by 100. Yeah. So if he reports a range that goes from -2 to -8, that's -200 to -800 kilopascals. Also remember that the unit is a small k for kilo, and then pascals is big P little a. All right, no other combination of letters. No big K's, no small p's, no big a's. And when you are doing this, this is the sort of bit where you've got to be pulling out values and using secondary referencing. So you might say, I'm not going to go in there and find the names, but you might say, you know, A and B, and then say, you know, 1941, cited in Kaczynski, 2012. Found water potentials of so much. Yeah? And with that, you have to acknowledge that they use different techniques as well. So in here you can acknowledge differences. You know, they use this technique, they use that. Also in this section, you need to say, and actually I probably should have put it up with that first bit. When you identify the isotonic point, explain what is that. It's where water is moving in and out of the potato at the same rate, so there doesn't appear to be a mass change. And at water potentials above that, something happens. And at water potentials below that, something else happens. Yeah? Yeah, I think I'd put that in here and say, you know, from the graph, the water potential of potato appears to be about -680 or something like that. This is the point at which water is moving in and out of the potato tissue at equal rates. At water potentials above this, water moves into the potato and its mass increases. At water potentials below this, water moves out of the potato and its mass decreases. I think I would put in that bit. So I'll put explain on there. All right, you will, hopefully, if this records properly, of course, have this on YouTube to check that too as well. So, bonus, yeah. So, yeah, having identified that, it is a matter of then sort of explaining what that actually means. You can use words like hypotonic and hypertonic in there if you want to. So that's the sort of analysis of your results against these ones that are reported in Kaczynski. I think this year's came out quite low, didn't they? You know, minus 600 and something is really sort of quite far down the range. A lot of them are a bit higher than that, but that suggests that the potatoes are a bit more concentrated, a bit less watery. And given the fact that we had a really dry summer, that's probably not surprising. So, you know, that's something to consider later, if you like. So you do the analytical bit. You then, you don't use a lot of bullet points and sections in here. You explain about the mean and the standard deviation. So mean, you need to say what it is, right? If you don't know, look it up. But also talk about accuracy. There were going to be 47 pieces of data. We ended up with 42, didn't we? Because I think we only ran 3 groups instead of 4 in here. So, yeah, so accuracy is improved by having 42 pieces rather than you just doing 5 of your own. So increased by number of data pieces. But also in here, you can say a bit more about the anomalies and refer to your appendix if you've got one. I would recommend it. So we remove anomalies because they are outside what we would consider to be, you know, an acceptable range. And then the standard deviation is, it shows spread, it shows precision, spread of data. So the variability of it, and the lines round about the isotonic point are reasonably small, so that's not too bad. What else do we need to do? We also need to sort of evaluate, you know, how well do we think it went? So comment on aspects of procedure. You could comment on, you know, compare with some of the ones that are in Kaczynski. But then other stuff that you'll find in there, factors affecting the potatoes, things like weather when they're growing, storage, how long they're stored for, and then things like varieties of potato, solutes that are used. Oops, that's not supposed to say variables. Varieties. Solutes, that sort of thing. And then I would suggest a very brief little conclusion, which is really that the results supported the hypothesis. And say, you know, we did find a point and it seems to be acceptable in comparison with other people's data. At that point, if you want to put a word count in, you can, but I'm pretty sure you'll all be overshooting anyway. You then put your references in. And as far as references are concerned, I am looking for at least one citation in every paragraph. Right, and on every image. So every paragraph you write, you know, 10 lines, let's say, there needs to be at least one citation in there. I want to know where that's coming from. The introduction should definitely have citations in it, yeah, because you're writing about these transport processes and the structure of the membrane, so where are you getting that information from? Yes, I know you know a lot of it now, and some of it will come from class notes maybe, but if you can find it from a reasonable source, could just be a textbook, that's fine, but I want to see at least one citation per paragraph. Sometimes you'll have a couple of lines, I can't put a citation in there. Think about it. It might be for things like the mean and standard deviation. You might find it's hard to, I'm not absolutely going to pin you down on every one, but most of your paragraphs, let's say, should have at least one citation. You need to be saying where stuff's come from. Now, everything that appears in your work as a citation must be listed in your references at the end, and everything in your references at the end must have been cited. All right, so those are the same thing. Remember, the citations are just little flags to tell you where to look in the references. So everything you cite must be listed so I could find it, and everything that's in your references must have been used. The appendix, I would personally recommend putting the spreadsheet in with the anomalies highlighted. You might need to shrink it down to fit it on the page, but it's not huge this year. So things that could go in there, you could have your spreadsheet of the full data set, but you don't want that in your work. You can use that dotty graph for finding the I don't know what we call it, points graph, to find the anomalies. If you want to put anything else in, you can do. It's largely up to you. Now, are there any particular questions about any of it that I can answer generally to the class? No? Okay. Use the guidance sheet, because it does tell you which assessment criteria you're hitting in which parts. With Kaczynski, don't be too bogged down by his beginning bit that describes the theories of osmosis. There is a section in there that is just about the water potential of white potatoes, I think it's described as. And that's where you've got all the data, but it does describe different people using different techniques and all that. So acknowledge those, because that, may give you different results. And then the end part, he's got a section of references, and then at the end there are some other bits and pieces that might or might not be useful depending on how many words you've got left, that do comparisons of things like the different solutes and that sort of thing, because he actually set that up as a guidance for a practical for his own students. So there is quite a lot there, but if you find it a bit overwhelming, just target in on that bit that's about water potential of potato tissue. All right. Okay, that's all the help you're getting on that now. Okay, what are we doing next? Hormones. Yesterday we made a small introduction to this unit by talking about homeostasis. And homeostasis is just a theme that's going to run through everything in this unit. Whatever system we're looking at, we're going to be considering homeostasis. So we're going to start off with two systems that are responsible for overall control of most of the processes in the body. And we're going to start off with the endocrine system, so that's hormones, and then we go on to the nervous system. So those two are the control systems, and then we look at circulation, respiration, and the renal system, so that we've got an overview of some control that's going on. So every now and then we'll sort of be looking at specific examples of control, and remember that's all about homeostasis, about maintaining constant conditions in the body, whether it's blood glucose levels, body temperature levels, blood pressure levels, 2 of those come into your possible assignments. Whatever we are controlling, it's going to be homeostatic. Now, the endocrine system comes to you as a PowerPoint. So you've got 3 sides with 12 slides printed on there. I'm not sure that that is all that there are, but you will find the original PowerPoint on Moodle. So you can run that in conjunction with these if you need to have a look at it. And of course, those who aren't with us this morning, that's what you do. You go on to Moodle, you download the PowerPoint, you watch the PowerPoint, and mind you, I'm going to be running it now anyway, but you've got that as an additional version of it. So let's find the PowerPoint, endocrine system. And I've got 13 slides on here, so I've obviously removed one from what you've got. Don't know which one yet. And we're going to just start by considering what it does overall. Endocrine, what does the endo bit mean? Inside, yeah. Endo is inside, whereas exo is outside. Now, endocrine means that the glands that are involved in the endocrine system, they secrete internally into themselves or into the blood that's passing through them. They do not secrete substances out of themselves into channels or tubes. So that means there is another set of glands in the body which are called exocrine glands, and they do secrete out into tubes and channels. For example, salivary glands into the mouth, sweat glands onto the skin. They've got tubes that come out onto surfaces or into cavities. But endocrine glands don't do that. They secrete into the blood that passes through them. The overall jobs of the endocrine system fall into these three sort of areas, really. We've got homeostasis, that's our biggie. And so the endocrine system is responsible for maintaining composition of body fluids. And that is bigger than you first think. You know, it is, as far as we're concerned, things like water levels, glucose concentration, pH, but it's absolutely everything about every fluid, you know, blood, tissue fluid, and anything else. And the endocrine system is controlling the composition of those fluids because that's what our internal organs live in. So it's really important. It's really controlling our internal environment. Secondly, it works with the nervous system to respond to stress. Most people are familiar with one particular hormone associated with stress. Well, cortisol, yeah. Adrenaline is the one I always expect. Yeah, adrenaline is what we consider the stress hormone, the flight or fight one. Yeah. And the effect is, you know, we tend to think it kicks in quite quickly, but actually it doesn't. If you're frightened by something, or you hear a loud noise, or somebody jumps out on you, that's not adrenaline that makes you jump, because hormones can't respond that quickly. That's your nervous system that kicks in first. And then if you need to maintain the stress level, the nervous system actually kicks off the production of adrenaline, and that's a long-term thing then. And this is the thing about hormones. And that's really obvious from the last point there, that they help to control growth, development and reproduction. And those are things that we're not really going to consider. But these are all really long-term, aren't they? Growth, you know, we're talking years and years and years. Development, again, years, and reproduction, months into years. So hormones work on this sort of scale. Hormones are much slower in their action, and they're much more general in the body as well. Nervous system, you know, it's very direct, it's very rapid. We're talking fractions of seconds. Whereas with hormones, we can be talking hours, months, years, you know, very long-term changes. So the two systems are going to work together to give us control that can go from milliseconds up to years. All right, so between them, you know, we've got a good sort of range of control. Most of what we're going to look at is in that homeostasis section, so not surprisingly. And I'm going to look at a couple of examples. We'll look at the water levels and the glucose concentration. We'll do the glucose one in a very simple way. Water levels also in quite a simple way because we come back to that in the kidneys. I might fit another one in, but I think, you know, those two will be plenty to give you an idea of how it works. Okay. Looking at the next slide, what we've got here is really showing us, you know, about this definition of the endocrine gland, that it secretes internally into the blood that's passing through the gland. This isn't any particular gland, but these blocks, these are cells within the gland here, and the little black dots on there are the hormone that's being secreted from the gland into the blood that's passing through. So blood coming in at the top appears to have no hormone in it at all. Sometimes that'll be the case, sometimes it might just have low levels. The cells are then producing this hormone, the little black dots, into the fluid that's circulating around the cells, and then from there those can pass into the blood, and so the blood that's leaving the gland has a lot more hormone in it, and that is how it works. So we're not getting this produced out through channels into any cavities, onto any surfaces. It's just as blood passes through these glands, it picks up hormone if it needs to. And there may well be a detector system in the gland that detects whether hormone is needed or not. Right, I was going to say there are a lot of these, but we'll have a look at those later. Sort of right at the end of this particular PowerPoint, I've got just an overview sort of map of the major endocrine glands, and then what some of the hormones do. But it's a very, very small snapshot, because actually there are hundreds of hormones in the body, but we only usually consider perhaps 20 of them we talk about. But it's not always obvious glands like the adrenal glands and the thyroid gland. There are hormones that are actually produced by the tissues themselves. So, you know, there are a lot of endocrine secreting cells around that are not actually glands, if you like. So we're only going to sort of look at some specific ones. Right, so let's assume that we've got a hormone produced by this gland here. How does it know where to go to? Well, it's the same as, you know, when you think about, right, I've got a headache, I take some painkillers, how does it know to go to my head? Well, it doesn't, does it? It doesn't just go to your head, it goes everywhere. But of course, it's only going to have an effect where you feel the pain. And in fact, most of the effect of painkillers is in your brain anyway. It's not actually at the point of pain. On the other hand, of course, if you take something that's anti-inflammatory, so something like aspirin or ibuprofen, they will reduce inflammation. But how do they know to go to the place that's got inflammation? Receptors. It's the important thing with understanding a lot of biological systems is those proteins in the cell membrane that we were talking about in the last unit. We talked about them being transport channels, didn't we? We've got the facilitated diffusion channels, we've got active transport proteins, but there's an awful lot of those proteins in the membrane that act as receptors. So that enables communication in the body. And these receptors, being proteins, have very specific shapes. So different cells carry different receptors. And the reason that they carry these different receptors is that the DNA doesn't know, but DNA in particular cells will be expressing certain genes that produce those proteins that make those receptors. So if you've got a cell, I can't think of a good example now, let's say in muscles, it will have insulin receptors. But a cell in your skin probably won't have insulin receptors. All right, so although the DNA, you know, has code for making insulin receptors, the gene for making insulin receptors is not expressed in skin cells, but it will be in muscle cells. So different cells have different receptors. Now, these receptors are shaped to fit particular chemicals. And in the case of the endocrine system, of course, they have to be shaped to fit the hormone. Now, if they have the correct shape, how do we describe them? It was one of my words. You'll remember it. Stereospecific. Yeah? These receptors, and I haven't put it on here, I've just said they carry matching receptor molecules, and we would describe them as being stereospecific. I want to write that on this slide. We know what happens if I touch the board when I've got this on. I have to click on there, get a pen. Oh, let's make it black just to be consistent. All right, so this idea of matching receptor molecules, all right, what that means is that they are, and I'm going to use this word more than once in this unit, this idea of being stereo. Stereo means shape, remember that? and then specific, so it means shape specific. I did it without making any mess. So they're stereospecific receptors. It's not that the hormone is stereospecific, all right, that's got a particular shape, but the receptors are stereospecific. So up here on the left, you can see a secreting cell that's producing hormone molecules, which are little blue circles. That circulates in the blood until it arrives at some cells down here at the bottom that say not a target cell, and the reason it's not a target cell, it doesn't have the receptors. And then we've got our two target cells over on the right there that have ridiculous receptors that look like they're sticking out from the surface of the cell. They don't, of course. These are the proteins that are embedded in the cell membrane, so they're part of the structure. But the two cells that we've got there have got these little green structures that represent the stereospecific receptors with little rounded areas on the end of them which can receive the blue molecules, and they fit in. So they can't, this hormone can't affect the non-target cell, but it will affect the target cell because that's got the appropriately shaped receptors on it. Now, moving on from this, we then have to identify two different sorts of hormone. And they work in slightly different ways in terms, first of all, of how they get, well, how they cause a response in the cell, or how they get into the cell, I suppose is the best way of looking at it. But the first sort don't get into the cell at all. And the two types are based on different chemical or biochemical molecules as their sort of basis. And what we have, which we're coming up to on the next couple of slides, we have what are known as, well, up at the top there you can see protein-derived hormones. I very often refer to those as peptide hormones. Now, there are a lot of different forms, but essentially they're built from amino acids. Now, amino acids are the building blocks for proteins, so you could say protein-derived or peptide, but this is coming up on the next slide, so I'm not going to worry about trying to describe it on here. Protein-derived hormones, they're the green blobs on here, which of course you haven't got colour on yours, so this is why it's worth having a look at it online, or on Moodle, I should say. But these green blobs, they fit into stereospecific receptors on the outside of the cell. They don't get in. All right? They actually trigger a change just by doing this. Because these proteins actually go through the membrane, they are then coupled to a sort of knock-on effect inside the cell. And we've got space, I've got a blank slide coming up that we can draw in, or you might need more space than that, but show you how this works. And it is a mechanism known as the second messenger effect. The second messenger is the effect, you know, what's actually produced inside the cell. The hormone is the first messenger, but the hormone doesn't get into the cell. It then triggers the production of a second messenger. So we'll look at that as a mechanism. So that's the protein-based ones, the peptide hormones. They just stick on the outside and effectively throw a switch. On the other hand, we've got the ones down here at the bottom right. We've got steroid hormones. And steroids are related to fats, lipids. They're produced by the smooth endoplasmic reticulum. And they are somewhat different because, being related to fats, of course, they can dissolve their way through the phospholipid bilayer. So if you're looking for an example of something that can just diffuse by simple diffusion into cells, then steroid hormones are a good example because they are soluble effectively in the phospholipid bilayer. They can just move through. They're non-polar. And this means that they actually get into the cells. So they will have an effect by getting in. Most of the examples of steroid hormones are reproductive hormones, sex hormones, things like estrogen and testosterone. And once they've got into the cell, they're then picked up by a receptor. The receptor isn't sticking out on the outside, really. They diffuse in. They're immediately picked up by a receptor. These look like little ice cream cones. That forms a complex, and then the receptor delivers the hormone to the DNA in the nucleus. Now, when we get to the final unit on the course, and it's not that far away, we look at the genetics unit. Or is it in the genetics unit? Or it might be in the biochemistry unit. Can't remember where I've put it. But we have a section on the control of gene expression. You know, how does a cell know which bits of DNA to use at any given time? One of the ways is hormones. And estrogen is a really good example. It's the example we'll use. Estrogen binds to the DNA and basically says, right, we need this gene to be expressed at this particular time. So it is it's a very particular little DNA switch. So we're going to look at those two types of hormone in a little bit more depth. We've got 4 slides coming up that give you a little bit of description and then a blank one for some diagrams. So we're going to draw a bit, and we can sort of get a comparison between the two. Okay, why is that not? Okay. Starting with the peptide hormones, then, or protein hormones, there's various ways of describing them. It's just lumping together several different groups of hormones here. But they're based on amino acids. Now, amino acids are something that we will meet when we're doing the biochemistry unit. But essentially, they are all organic acids, and we've just started the organic chemistry. But they all have a little group in common. They all have, apart from the acid group that they've got, they have an amino group. And the amino group is a nitrogen and 2 hydrogens. So it's an NH2 group, is an amino group. So they've all got that. And they're used for building proteins. So all proteins are built out of amino acids, anything from a couple up to hundreds. And so what we've got here, we could talk about peptides. Peptides are usually small chains of amino acids, whereas proteins are generally bigger, I suppose. That's the way we describe it. They're water soluble, as it says here. So, we might expect they're not going to get through the membrane. And they vary in size from thyroxine, which is only 2 amino acids long, to growth hormone, which is 190 amino acids long. Now, just a bit of a sidetrack here, because I've sidetracked for a while. A lot of hormonal disorders can be treated very, very simply by giving people a hormone that they're missing. You know, it's not complicated drugs that have to interfere with processes or anything like that. You know, if somebody's short of a hormone, you can just give it to them. But there are different forms of delivery, aren't there? So, for example, I mean, we've got growth hormone there. If somebody needed growth hormone, they'd have injections of it. However, I take thyroxine every day, but I take tablets. So why can't the person who needs growth hormone take tablets? It's to do, and this is making an assumption that you're sort of thinking about the digestive system. If you put anything that's more than a few amino acids long into the digestive system will, digest it. Yeah, it's not just dissolve it, it's actually digest it, break it down, and so you've no longer got the hormone. So if you were to give someone tablets of growth hormone, or the same thing, of course, with insulin, that's why people with diabetes need to take injections of insulin. I can't remember how many amino acids there are in insulin, but there's 2 chains, and there might be about 50-something, can't remember. But if you gave somebody insulin tablets, they'd just digest it like a protein, and it wouldn't get into the blood and it wouldn't do its job. But thyroxine is so small, 2 amino acids, the body doesn't bother digesting those because they're small enough to go straight into the blood. So, as far as I'm concerned, that's a benefit. So I don't have to inject thyroxine every morning. I can literally just take a couple of tablets and it keeps me alive. But hormones are very, very variable in that sense. they don't actually get inside the target cells. So the important thing here is that they bind to these stereospecific receptors that are embedded in the membrane and are the right shape to fit these particular hormones. And this triggers the chain reaction, and what you get is actually what we describe as a cascade effect. So you might get one molecule of a hormone, like adrenaline, binds to a receptor and then triggers the production perhaps of 100 second messenger molecules, and then each second messenger might trigger the production of 1,000 other molecules. So you get a sort of multiplying effect. You don't need very much hormone for them to work. Examples that you could put in here, or if I'm going to write them in, I need to click on the pen, don't I? So that one. Let's have red this time. Well, I've mentioned some. So what should we put in as examples? I've just mentioned a couple. Straight in one ear and out of the other. Insulin. What else have I mentioned? Whoops. Adrenaline. There's one up there, and we can, oops. Yeah, thyroxine, that's supposed to be adrenaline, which has an E on the end. I'm not being careful enough on this one. Yeah, well, let's hijack those two. Thyroxine. Growth hormone, yeah. Yeah, are you paying attention? Yeah, there's a very strange effect on this board. So the majority of hormones are actually peptide hormones. The majority of them don't get into the cells. They do just bind to the outside. And, this is actually really useful then for other drugs that you might want to use to interfere with the effects of some of these. And a particularly good one is beta blockers. Heard of beta blockers? Yeah? What do beta blockers do? They reduce something. Well, they reduce stress levels and heart rate and blood pressure, really. They're used for heart rate and blood pressure if it's got a stress component. What hormone's concerned in a stress reaction? Adrenaline. And what beta blockers do is to block the adrenaline receptors on the heart. So adrenaline can't then attach to the heart, so it reduces the heart rate and the blood pressure by stopping adrenaline from attaching to the heart. So how would you design a beta blocker? Well, you look at the shape of adrenaline and you make something that is similar in shape that will fit into the receptor without triggering a reaction. Obviously, there's no point in making something so similar that it actually sets off the same process. But to block a receptor, if you've got a receptor, let's say, which is, I'm just going to use a bit of this space. If here's the membrane and the receptor is, let's say, triangular like that, because the hormone is that shape, and that will fit in, if that's your hormone, well, you don't have to have a drug that's exactly that shape. As long as you've got something that will bind in there, it might be something sort of shaped like, I don't know, like that would actually block that space and stop the adrenaline from getting in. So that might attach it. So I've made that rather small, actually, but that might be sufficient to block the receptor. So you just have to find the right little chemical sort of tags, if you like, round the outside of the molecule, perhaps round here, which is where it would sort of bind in here somewhere. It doesn't like me doing dots, does it? But the idea is that if we know something about the shapes of these hormones, then we can stop their action as much as giving somebody extra ones to replace them. Another example of blocking. Well, there's actually several of the blood pressure drugs are blockers of some sort. So you've got beta blockers. You've also got ACE inhibitors, if you've ever heard of those. If you know anyone who's taking blood pressure medication, if it ends in pril, so things like ramipril is a very well-known one. that blocks, it's actually an active site on an enzyme, which is sort of the same job. And then there are another group called angiotensin II receptor blockers. And again, that's a hormone. And the receptor blockers bind to blood vessels and stop the hormone getting in that will squeeze the blood vessels and make them tighter. And so we'll actually come back to some of those, because if you choose to do the blood pressure assignment, then you will need to look at how beta blockers, ACE inhibitors, angiotensin receptor blockers, how they interfere with the normal control of blood pressure to actually, get it back to normal. So we'll just keep throwing a few of these examples in. But so these are all then binding to the outside of the cells. They're not getting into the cells. And what we now need to do is to look at how these actually do their job. How do they work? Right, I have a dot. Oh no! Yeah, that's what happens when you use the rubber. I've got a rubber option. Eraser. Oh, I had one that said erase all incoming slides. Right, let's go back to, whoops. Let's go back to some drawing. It's really annoying because I can't just pick up different coloured pens on here. So I'm just going to have to choose some colours. But you might feel you need a bit more space than what I've given you as the printed version of this. You've got a tiny little box down there. You might want a bit more space. But this is looking at how a peptide hormone works. If this is a bit of cell membrane with a receptor protein in it, I have to draw quite slowly because we do get these odd effects. Right, so this receptor protein has got a round sort of space in it. So let's start by labelling that. That's our stereospecific receptor. And let's have, I have to change the colour on here, don't I? Can't just pick up the different coloured pen. Right, let's choose blue. It doesn't matter what colour pen I pick up. If our hormone is these little things, then they can come along and bind themselves into the receptor. Right, so this represents our hormone. The hormone doesn't get through the cell membrane, remember, it binds, and then it'll come away again. It'll detach, it'll dissociate from the receptor. But when it binds to the receptor, it triggers processes in the cell. Now, I'll sidetrack here onto the example if you're going to do the blood sugar assignment. With insulin, when it binds to the insulin receptor, the effect of that is to open facilitated diffusion channels for glucose. All right, so that's what insulin does. Once it binds to the receptor, it's responsible for opening the gates, if you like, so that glucose actually gets into the cells. All right, there's several different sorts of glucose transporter, but the ones that are really important for us, looking at the hormonal control, what the insulin does is to open those channels. But generally speaking, what happens, that's not a direct thing. We still have to look at our second messenger effect. Now, let's choose another colour, just bear with me. Oh, it's that green. The effect is that the binding of the hormone to the receptor triggers the production of an enzyme. And the enzyme is called, it has slightly different names in different sources. It's called adenyl or adenylate, sometimes, cyclase. Now, that adenyl bit, adenyl, Heard of that in... Well, ATP, the first name, the A in ATP is adenosine, and that comes from a base called adenine, and that's what this is about. It's about that same base. You find it in ATP, you also find it in DNA, so it's a very useful substance, and it's part of our second messenger system. So this is something to do, it's actually to do with ATP, to be honest. Cyclase, well, that sounds like a circle, doesn't it? A cycle. And what this enzyme does, so let's label it an enzyme, is that it starts with ATP. I'm not going to change colour again because it's just getting a bit ridiculous. And it converts ATP. I think it starts with ATP. Now I've said it, I'm not convinced. I think it does. It converts it into cyclic, not surprisingly, AMP, which is adenosine monophosphate. So whips a couple of phosphate groups off, wraps it round into a circle, and this sometimes abbreviated to just a small c with AMP, so like that. And that is our second messenger. Cyclic AMP is the second messenger. And it's that then triggers further responses in the cell. Right? Curiously, it triggers whatever response that cell wants to produce, but obviously the control is a lot more complex than this. So I'm going to go for one more colour. We'll go for purple. All right, so this is our second messenger. And this will have the effect, whatever's going on. So this is where you get this cascade effect. So you actually get a multiplying process going on here. And to give you an example that I haven't mentioned yet, I've mentioned insulin, and that lets glucose into cells, and it can be stored in cells by joining it together into big chains called glycogen. However, when blood sugar levels drop and you need to get more sugar out again, that glycogen needs to be broken down and released back out. And you can there are various hormones that will do that. There is a specific one in that system called glucagon, but also adrenaline will help you release sugar into your blood, won't it? So that you're ready for action. So either of those hormones, one hormone molecule binds to the receptor, let's say adrenaline. That triggers the production, let's say, I have no idea what the number is. Let's say it produces, I don't know, 100 molecules of adenyl cyclase. 100 molecules of adenyl cyclase might result in the production of 1 million molecules of cyclic AMP, and then each one of those might trigger another 1 million conversions of glycogen into glucose. So at each step, you can increase the effect. So you only need small amounts of hormone to actually trigger quite a large response. So they're only present in the body, these compounds, in very, very small amounts, because we just don't need a lot of them. So that's the peptide hormone then. No involvement of the nucleus or the DNA. We've not got anything sort of deep going on there. It's not affecting the genes that are expressed or anything like that. But we've got a switch being thrown, a chemical switch, where the hormone triggers the production of an enzyme. The enzyme triggers the production of the second messenger. The second messenger has the effect. So that's our peptide hormones. Okay, we'll have a look at the steroid hormones now, and then, we haven't got a lot left on that. Should neatly fit in before break, I think. Steroid hormones are somewhat different, and people have usually heard of steroids in certain applications. I mean, what sort of applications have we heard of steroids? Well, sport or not sport, isn't it, really? Abuse in sport, let's say, yeah? Now, the steroids that are usually abused in sport are very often male hormones, because those will increase the body's ability to produce muscle if somebody is training really hard and making a demand for muscle and also eating a high protein diet. You know, you can't take your steroids and sit on the sofa eating steak and build muscle. But similarly, you know, you can't take your steroids and do hours and hours at the gym without putting the proteins in. So you've got to have this combination. But we know, you know, they are very dangerous. They have a lot of unwanted side effects. But yeah, so they're abused in sport. But there are also medically used ones, aren't there? So they're used for inflammatory conditions very often. So for things like asthma inhalers, there's two, aren't there, asthma inhalers? You have I can't remember which one's which, but there's a steroid one for, you know, if there's an inflammatory sort of process going on. But you have the one that just dilates the airways, and then the other one is a steroid one. Plus steroid injections, sometimes into damaged joints, or steroid creams for, you know, inflammatory skin conditions. So we've got a bunch of hormones, but not very many that fall into this category. So these ones, because they can actually get into the target cells, you know, remember they're diffusing through the membrane, that's where they bind with their receptors. And they have a much more, I don't know what the word is I want, but, you know, a much greater effect within the cell. They're actually targeting things themselves. So they actually bind to the DNA. There's actual sort of connection between the hormone chemical. You know, one molecule of a steroid can attach to one molecule of DNA. So, but then again, that's all you want, because the one molecule of DNA is where the gene is. In a given cell, of course, you've got 2 copies of every gene because you've got pairs of chromosomes. So there's no point in producing, you know, having, well, you don't, you know, there is no cascade effect going on here. One hormone molecule attaches to one of the chromosomes, another one might attach to the other chromosome, but that's not necessarily even going to be working. So yeah, this is it's quite a different sort of process. But the sort of examples that we've got here are, well, the reproductive hormones, as I said. So we've got estrogen. The other female one is progesterone. I'm sure you know that. And if you're not answering questions because you think they'll be heard on the microphone, they won't. So anybody listening to this, if you sense gaps where you can't hear anything, it's because people are actually answering questions, although they're not this morning. So estrogen and progesterone are the female hormones that we're familiar with. And then, of course, testosterone. Yeah, there you go. Testosterone. And there is another one, and that's cortisol. And that's the one that's used for the inflammatory responses. Cortisol, you've probably heard of hydrocortisone cream. Yeah, that's based on cortisol. That's the anti-inflammatory one. And that's the basis for, you know, the anti-inflammatory inhalers. And people who've had transplants very often have to take steroids, don't they? And they'll be based on this one. The reproductive hormones, you can tell they're steroids from their names, because they have a steroid, steroid, and then a shortened version. So we should have estrogen. Of course, if you see it written in an American source, it won't have the O at the beginning. It'll be estrogen rather than estrogen. But we do have some strange spellings in English, but I like them. So here's our steroid hormone examples then. So we need to illustrate these in the same way as we did with the peptide one. Let's start again with a black. This time I'm going to need a nucleus in here. So we will have our cell membrane. Although I haven't put a receptor in it, the receptors will probably be in the membrane, to be honest, or very close to the membrane. So I'm going to put the steroid receptor let's make it a little circle this time with a triangle in it. There's our receptor. And then we need a nucleus over here. Oops. And then just do DNA as a wiggle. A double wiggle. I suppose I better label it because it's not that obvious what that is. Okay. Change my pen colour. This time, what colour should we have for our hormone? Let's have an orange hormone. See if that works. Okay, here's our hormone. Should be the little triangles. So we don't need much of it, obviously, but there's several of them. And we've had one get through here. So hormone. Oh, yeah, I need to go. I'm going to have to go back to black. I'm actually only going to need the orange and the black. I'm going to just fill in the hormone while I've got the orange pen turned on. It's going to be transporter. I'll have to attach the transporter to this, but it's going to be carried over here. It's then going to be sort of dumped into the nucleus, and then it will bind itself to part of the DNA. Let's tuck it in one of the little folds there. Now, if I go back to the black, this will make a little bit more sense. If I could find the pointer, I think it's gone there. Back to that. So our receptor has got a lot bigger. It takes the hormone into the nucleus and lets it go. Like Pac-Man, and then that's really all we need, to be honest. It's a much simpler diagram. You know, we haven't got a second messenger to explain here, and the let's just describe what the hormone's doing over there. I don't work well when I've got too much choice. Give it a blue. So hormone, what it's doing here is that it acts as a gene regulator. So it switches genetic activity on and off. Well, on, basically. So the use of this for you in terms of assessment, because it's always worth mentioning that all the way through, is really through the assignment. I very rarely put hormones in the exam because they've pretty much been covered by the assignment. For the blood glucose assignment, you've very obviously got the effect of insulin, but as I say, it's got a partner hormone, glucagon. Adrenaline could be mentioned in there as well, but you know how they work is by binding on the outside. They're peptide hormones, and it's that opening of glucose channels that's really important there. With the blood pressure assignment, the hormones that you've got there, well, adrenaline again is one of them, and these other hormones that can do things like making blood vessels constrict to increase blood pressure. And so it's, you know, sometimes people have high blood pressure because the blood vessels are sort of, you know, too tight, if you like, too narrow. And so medication can be used to stop those hormones having their effect. So you just get other substances to bind and block that access. So you're not really going to need the steroid ones. They're just there to complete the picture. But they will come back to us when we're doing gene regulation. So, you know, these don't necessarily have a use. Well, they don't have a use in the exam. But they will be important in the assignment for this unit, but also the steroid side of it. We'll explain that in a lot more detail when we get down to either the bit on protein synthesis or the bit on genetics. Can't remember where I've put it. I moved things around a couple of years ago, you see, so yeah. Just trying to remember where I wrote the new bits in. Right, we haven't got very much left to finish on this, so I think I will just continue on and finish the last few slides. And then what we'll do after break is to start having a look at some questions and activities, because with the exam at the end of this unit, it's the first proper exam that you'll come across. And it's, you know, getting used to using information to answer questions rather than researching stuff for assignments is a different skill. So we throw questions in right from the word go on this one. So I've got some various little activities that we'll have a look at after break. So let's finish these off. We should have I think there's 3 to go. Right, this is a map of where these endocrine glands are in the human body. And, well, this is a selection of, you know, and like I said, there are a lot of structures in the body that actually produce hormones. We, you know, might refer to them as endocrine cells. Sometimes they have other names as well, but, you know, there's a lot of stuff going on that doesn't actually involve specific glands. Endocrine glands are, you know, how do I describe them? Discrete structures where you've got the hormone secreted into the blood that passes through them. But then you have the other ones. The tissues really are just secreting into the area where it's needed. And we're just going to look at where these are, some of the key hormones and disorders. And again, it's really only looking at the highlights as far as the hormones are concerned. If you look up hormone tables, you'll find that whereas I might identify 2 hormones from one particular gland, you can find a list of about 20 slightly different versions. One's converted into another one, or an inactive form and an active form, and yeah, all sorts of things like that. But I just need to sort of, you know, keep it minimal, and just sort of, you know, we're looking at an overview. Oh, the other thing I'll do after break is to, yes, I'd forgotten, to go over a couple of examples of control systems, just to give you an idea of how the homeostatic mechanism works with those. Okay, if we start at the top of this now, you've got, yeah, so you've got this slide. You've also got, it looks like the next one is talking about the connection between the nervous system and the endocrine system at the pituitary and hypothalamus. I'm probably going to divert out. It's a bit easier for you actually because you've got them all on one page. I'm going to go backwards and forwards between the slides, I think. You've also on the final slide got some of the main hormones. So I'm not necessarily going to use that slide. I will talk through the hormones as I talk through the glands. So, yeah, not going to stick directly to the PowerPoint, but I will use this plan. So if we start at the top, we have the pineal body or pineal gland. Now this is sort of embedded sort of in the depths of the brain, and sometimes known as the third eye, it used to be. All right. Reason being is there is a response to light. Now that's because light falling in the eyes has an effect on the pineal gland. And it's something that wasn't actually on the list of endocrine glands when I first started teaching. You know, it's appeared in the last 20 years, it's on every list now. But the hormone that's produced by the pineal gland is one called melatonin. Now, melatonin's got nothing to do with melanin, the colour in the skin and hair, but melatonin is associated with sleep. Yeah, that's the one. Melatonin, you can actually take melatonin to try and increase your sleep. I've tried, it didn't work for me. But melatonin is produced in the dark, or melatonin is destroyed by light, or not entirely sure what the relationship is, but basically you get more melatonin when it's dark and less when it's light, and melatonin makes you sleepy. Consequently, on days like yesterday, well, it really didn't get properly light yesterday, did it at all? Today looks a lot brighter out there. But yesterday, I could quite understand if people just felt tired and sluggish and a bit low, because the daylight wasn't sufficient to break down melatonin or stop its production enough, really, to let people feel properly awake. Now, this has some real impacts in certain situations. So there's a condition where some people produce far too much melatonin in the winter and suffer from. SAD, that's the one, yeah. Seasonal affective disorder. Seasonal affective disorder is real. People produce too much melanin, particularly in the winter, but can be other times of year as well. Very often the best thing for them is to get out into light, but they don't feel like it necessarily. But in real serious conditions, people are, very depressed, start overeating. I mean, we all think, oh, actually, yeah, really dull days, yeah, I don't feel great and I do want to eat more. And a lot of people get what is sort of vaguely called the winter blues, and it's, not really a problem. If you think about our evolution, it would make us stay in shelter in the winter, wouldn't it? And trying to get us to eat, because it's perhaps going to be harder to find food then. So it has a purpose. But of course it can really interfere with, what you're trying to do on a daily basis. And so it can be treated with the lights, SAG lights. Yeah, special daylight lamps that, if you sit in front of that for half an hour in the morning, not quite. It's supposed to hit the back of your eye at a certain point. And so I think you're supposed to put it just above your eye line. And I did a lot of research into it because I got one for my eldest son because he seemed to have a real issue with it. There's a lot of evidence to show they really work, really make a huge difference to people. So melatonin, it's quite useful sometimes, but then it can be a real pain. The other time, of course, that it's a pain is jet lag. If your light and day gets all messed up when you're travelling around the world, then your body doesn't know whether it's night or day because you get all the artificial sort of light and stuff like that. And it's useful, but it can be a pain. Anyway, moving on. Next one, well, we'll put the next two bits together. The hypothalamus is actually part of the brain. It's not an endocrine gland in itself, but it does produce hormones. But it's part of the brain. And so the endocrine nervous link is between the hypothalamus and then that other structure over there which dangles off the bottom of it, and that's the pituitary gland. And if you have a look at the next slide, I will try and go onto that and then back. Yes, I can do that. Here is our brain with the hypothalamus at the base of the brain. And then dangling off the bottom there, looking like Rudolph's nose, you've got the pituitary gland. It is not that big, round and red. Right? But there is an absolute physical connection between them, which you can see in the right-hand diagram, where you've got blood vessels in this dangly grey thing down here is the pituitary gland, but then nerve cells in the hypothalamus are linked into these blood vessels. So you've got communication between the two at that point. And so, it's like saying, we've got these two control systems, endocrine system and nervous system. Now, they can't just work independently. And they don't. There is a little degree of crossover where you've got the pituitary and the hypothalamus. So there are certain substances, what they call neurohormones, that are produced by these nerve cells in the hypothalamus that get into the blood, go into the pituitary, and are then modified and released by the pituitary. So, they're working together at that point. Right, let's go back to that one. The pituitary generally is responsible for a whole load of hormones, great big list. But if you look at the table on the last slide, I've only given you a selection. In its own right, the pituitary, I've given you 5 there, mainly because that's all there was space for. Growth hormone comes from there. Thyroid stimulating hormone, that's not thyroxine. All right, that's the hormone that makes the thyroid produce thyroxine. Adrenocorticotropic hormone, that's a hormone that triggers the activity of the outer layer of the adrenal glands. follicle stimulating hormone, which causes the development of follicles in female ovaries, not as opposed to male ovaries. And then luteinizing hormone and prolactin, which are also sort of part of the reproductive hormone suite that we get in females. So that's just 5 out of lots. Down at the hypothalamus on that list, you've got releasing and inhibiting hormones such as, and this is the thing about the communication between those two. There's a balancing, switching on, switching off control sort of process going on there. Oxytocin is one, which again it's associated with contraction of the uterus. And then one that we are going to need, this is one, just put highlight it or make a note by it, antidiuretic hormone. That's one I will come back to after break and describe how it works, because we will need it again at the end when we do the kidneys. So it comes from the hypothalamus. It's got several names, but that's one. That's going to be one of our important hormones in this unit. Right, moving on from the pituitary, we come down into the neck, where we've got the thyroid gland sitting there like a little bow tie. And the thyroid gland produces a couple of hormones. Most important one, most significant one is thyroxine. It's sometimes called T4. And the reason it's called T4 is because it has 4 iodine atoms or molecules, not sure. 4 iodines of some sort, iodine atoms, let's say. There is also a T3, all right, and there's a conversion between those two. But T4 is what they often measure in the blood. And thyroxine is just responsible for maintaining metabolism in cells. It maintains the rate of respiration, so energy use, that sort of thing. That's really weird, because what that means is, we need thyroxine just to keep the cells ticking over, just to keep them living. And you think, well, why? That's putting another step in a system. Why do you actually need something to make it do what it should be doing? Because if you lose the thyroxine, then the cells basically just stop working. And if people have low levels of thyroxine, they become very slow and sluggish, put on a lot of weight, lose mental sharpness, all that sort of thing. And, you should be able to manage without a hormone to stop that. But that's what happens. And ultimately, if somebody went without thyroxine, they would just go into a coma and die, because their metabolism would just shut down. If you've got too much thyroxine, it goes the other way. Your metabolism speeds up, people lose a lot of weight, and actually thyroxine has been abused as a weight loss drug. But it's a really bad idea, because it also affects the heart, makes the heart go too fast, and causes a lot of heat production in the body. And you just think, what's the point of this? And in order to produce enough thyroxine, what's it going to detect? How's it going to know you need more or less? Well, it sort of has to be self-regulating. And this is where you have a couple of hormones up here that control it. There's the thyroid stimulating hormone that comes from the pituitary, but also the hypothalamus produces another hormone that sets that off, and then that sets that off. And they sort of have to look after themselves. Are we producing enough of ourselves? Oh no, we'd better make some more. Oh yes, we are, so we can cut that. It's the most pointless system, really. But there we are, we've got that. Sitting on the thyroid gland, we've got the parathyroids. They produce something wildly named as parathormone or parathyroid hormone. Makes a change to have something that simple. And that's concerned with calcium metabolism in the body. I never remember which way around it is, but we have hormones that will raise our blood calcium levels by taking it out of bones. We also have hormones that will lower our blood calcium levels by putting it back into bones. And estrogen fits in there somewhere, because you might be aware that after the menopause, women are much more prone to osteoporosis, which is loss of the hard material in the bone. So the bone is broken down more, and estrogen protects against that, and it will be the effect of one of those hormones. But that's what the parathyroids do anyway. They're helping to control calcium levels in a homeostatic sort of way. We then have this odd one here, the thymus. Now, this body is an adult-looking body, and actually, we really don't have very much thymus in an adult. The thymus gland is, I think it's just in front or behind the sternum, the breastbone, and it's very, very active in children, helping to mature some of the white blood cells. Now, by the time we reach puberty, we have encountered the vast majority of infections that we're going to meet in our lives. So part of the body changes in puberty is that the thymus gland starts to shrink. So we're not left with very much thymus as adults, so much more active in children. Working down, not many left. We've then come to the adrenal glands, and you can see they sit like little hats on top of the kidneys. And the name adrenal is ad, means on, and renal is kidneys. So adrenal glands, adrenal glands, they are sitting on the kidneys. They've got an outer layer called the cortex, an inner layer called the medulla, and the main hormone we're familiar with them producing is, of course, adrenaline. But if you look at the list, you'll see that they're responsible for producing cortisol as well, and they do produce small amounts of sex hormones. And what they tend to produce is the hormone of the other sex. So we all produce, females produce male hormones, males produce female hormones, and sometimes the balance is wrong. And sometimes, and the particular example really is where you get women who naturally produce fairly large amounts of testosterone from these glands, and it can really cause problems in sport. Because, of course, one of the things they test for is hormones, and it depends how much athletics you watch, but I don't know whether anyone's familiar with Caster Semenya. Yeah, I think she's South African, I believe, but she is a heck of a runner. She is pounding the opposition at, I think, 800 metres, thereabouts. But to look at her, you can see that there is something that, I shouldn't say isn't normal, that's an awful thing to say. But I think she naturally produces, and she was out of athletics for a while they did investigations, but they never released the results. But if you look at her, then you can pretty much tell there's more testosterone there than you expect, you say. It's a protective sort of thing. We should have a balance, and in every balance, it's sometimes not what we would expect, perhaps. And what does that leave us with? The pancreas. Well, pancreas has two jobs. It has got an endocrine role in producing hormones that control blood glucose levels. That's insulin, and the other one, glucagon. But it also has an exocrine role. It secretes out of itself. It secretes digestive enzymes out. So it's actually a gland that's a very, very busy and important one. So it's producing digestive enzymes to break food down, but it's also producing hormones as part of its endocrine function. And then we've got, you've got the, take your pick, testes or ovaries. I think I chose this diagram because it did at least have the decency to put them in separately. There are diagrams that have both in, and you just think that just doesn't look right. So ovaries in females produce a couple of hormones, estrogen and testosterone, no, estrogen and progesterone. Testes produce testosterone, fairly obviously, and these are just part of the control system for human reproductive cycles, producing gametes, that's the reproductive cells, and looking after developing embryos and all that sort of thing. So quite a variety of functions, and actually I think out of all the body systems, this is the one I like the least. And the reason for that is because they're all these disconnected sort of structures that aren't really working together and having the same job. I like the circulatory system where it's all working together to do one thing. Whereas here we've got, one of those, there's no real similarity, say, between the thyroid and the adrenals. And, I find this a bit bitty, shall we say. Okay, I think then that's covered this bit where you've got the connection between the pituitary and the hypothalamus, connecting the nervous and endocrine systems. And it's taken us through, those are, that's why there's an additional slide on my version. Those are examples of bigger hormone tables. and then we've gone through all those as we've talked through them. And I'm going to come back out of these, once we've had a break, I'll come back to a little bit about insulin and glucagon from the pancreas, and a little bit about antidiuretic hormone controlling our water levels. And then we'll have a look at some questions and see if you've learned anything. Okay, we'll stop at that point and take a break. Right, I don't want to keep those. Let's just